Hello, ladies and gentlemen. Welcome back. Nice to see you again. In 3, 2, 1. Ooh. Whatever. Welcome back to Flow from Hilton, and thank you for joining me. Once again, I say once again, but it's probably not even once again. I just started, so how could it be multiple times if I haven't even done it multiple times yet? So thank you for joining me during our inaugural series here on Clark and Harkin. Just kind of going to go against the grain, and today's topic is going to be the coronavirus. Whoop. Where did you pick that interesting and novel topic, you might ask? Well, unless you've been living under a rock, obviously this virus has had a severe impact on you. Hopefully, from just an economic standpoint, and you haven't had to deal with any of the health or physical aspects of actually acquiring the disease. But I thought I'd give everyone just my kind of comical take on the factual data regarding virus replication and the viral life cycle with COVID-19 as our example. So let's kick it off with the idea that there are several types of viral genetic components. Some can be DNA, some can be RNA, some are positively charged, some negatively charged, double-stranded, single-stranded, and all sorts of flavors in between. But the COVID-19 in particular is a positive stranded RNA virus. Its implications will be understood a little bit later in my explanation, but that's all we need to know about that for now. Viruses are very clever for how simple they appear to be. Just for comparative circumstances, the average density or gross number of genetic information or copies that the virus has is about 30,000. So 30,000 base units are what's on average used to create a working genetic pool within a virus that is successful in replicating itself and subsequent infection. Humans, on the other side of the scale, for example, we have about 30 billion base pairs in our genetic code. That's a little bit of a difference. If I could do basic math, I would do the percentage of 30,000 divided by 30 billion equals 10.1 million%. I don't know. I'm fucking stupid when it comes to that. If I thought about it for a second, I could do it, but my brain wouldn't. It doesn't want to focus on something. It's like negative. You're not going to get that answer. Or even in the ballpark range. So move on, buddy. Okay, back to viruses. The nuclear envelope, or it's like the cell membrane for the COVID-19 virus, is lipid-based, just like our own cell membranes. So the main route of infection is orally or nasally, but infection can also occur through the eyes, which is kind of fucking creepy and weird. That's why when you go to the doctor to get tested, and the doctor pulls out what looks to be like a Q-tip and starts nose fucking you with this thing, he's not some freak. He's trying to get way back into the nasal cavity, kind of where it connects to the throat, where the virus is typically found to embed during early infection and some of the early symptomatic phases of being infected by this virus. So, after it has infiltrated the host, it is target specific. So the COVID-19, I'm sure as you are all well aware of by now, is a primarily respiratory disease, and it affects our lungs and respiratory tract in a couple different ways. But that's the cellular specific infection for COVID-19 respiratory. And in particular, it preys upon these very, very susceptible cells that line the tiny projection-like surface of the inner lung that's involved in gas permutation. So these cells use a clever little trick involving iron to tease oxygen out of the atmosphere that you breathe across the alveolar cell membrane and into your blood supply via iron in hemoglobin. So that's another doozy of a tale, but we'll save that one for later and get back to our story. So kind of interesting. The nuclear envelope of COVID-19 fuses with the cell membrane of the host infected cell. They kind of become one, and it shoots its load into the host cell. Now, what's kind of interesting is that one of the first genetic sequences that it expels into the host cell is a base series that encodes a delay in the infected cell's immunity response. So it's basically trying to buy itself some time in order to replicate and assemble new babies, which are called virions, by the way, and eventually lyse the cell and release these new viruses into adjacent tissues for subsequent infection. Another interesting factoid, people are tripping out. Oh, it's the bats, man. They ate the bats. Well, the reason why this COVID-19 virus is associated with bats to begin with is there is a protein pathway that's utilized in this viral infection in bats using I believe it's the ACE2 gene. Down there in the comments, correct me if I'm wrong. I think that's what they say, these two bubers. Anyways, so we know that this COVID-19 virus that's been sequenced by the Chinese first also contains this same ACE2 series, so that's why we think it made the jump from bats to humans. Why is it named the coronavirus? That's pretty simple, stupid answer. Under electron microscopy, there are these small corona-like projections all over the surface of the virus, hence corona. The COVID-19, the 19 just has to do with the year 2019 that it was first sequenced and confirmed, reported by the Chinese. So what do we get to? Back to the story. So the virus has bought itself some time by releasing a preliminary sequence that delays the host's immune response, and after that, this is the clever part of this whole virus. It's such a simple organism, so it goes by that old monogram or whatever it's called, KISS, K-I-S-S, keep it simple, stupid. So it has gone through an evolutionary pathway that has decided that having its own cellular machinery to duplicate itself or replicate itself is not in its best interest, and it would be more efficient and evolutionarily successful as far as progeny are speaking, if it in fact took advantage of the host cellular machinery in order to keep its life cycle productive. So it's bought itself some time. Now it starts injecting all of these snippets of genetic coding that basically provide the instructions for the assembly and creation of new duplicate viruses in the infected host cell. So kind of neat. The host cell cannot determine or it has no bias towards the genetic information that it's decoding and eventually translating and transcribing. So is that the fucking neighbor, dude? Come on. So anyways, we are now going to shut this. There we go. So, excuse me, I'm having some microphone issues. Alright, back to the story. So it has assembled these new little baby virions. It rides this cellular machinery like it fucking just stole it. It just fucking, oh my god, dude. Ribosome, they're getting fucking just worked. You know, they are putting out so many new little baby components that they exhaust the cell, and the cell goes through a lysogenic phase where the cell membrane ruptures, the new baby viruses then flood out into adjacent respiratory tissue for subsequent infection. Some of these baby virions are condensed into small droplets and expelled during coughing fits, normal breathing, just during your day-to-day interactions. So that's kind of interesting. While we weren't talking about the nuclear envelope of the COVID-19 virus being lipid-based, kind of like most cell membranes, that's why all you got to do is wash your hands. You know, lipids are disrupted internally at the chemical level by, you know, by lipids, by a soap. Lipids are affected by soap. You get my picture. Wash your hands, motherfucker. It's as simple as that. That's why, you know, you don't have to have some crazy high level of alcohol, excuse you, Donovan, or any other sort of disinfectant, which I'm not recommending against. I'm just saying simple warm water and soap is very effective in destroying the virus. There haven't been a lot of long-term or robust studies as far as the life cycle outside a host, like how long will it last on particular surfaces. Usually the CDC will run tests that characterize how long and under what conditions viruses and bacteria are able to stay viable prior to becoming compromised and no longer able to infect an individual that comes in contact with it. So, how about a misconception, Eric? Sure. God, I got my finger on the pulse. I'm feeling you guys. All right. How about some misconceptions or some things we're confused about? Sure. What if I take these old antibiotics that I have? That'll fucking fuck it up and give it no chance. No, antibiotics and antiviral are completely different. They are two totally different ball games. Antibiotics will kill bacteria, not viruses. They're very, very distinct classes of biological entities that are distinct by nature. Therefore, taking antibiotics will have no effect on your viral infection, and vice versa. Taking antivirals will have no effect on a biotic. You get my drift, dude. All right, you get my drift. I'm being a little verbose, but you get it. You're trying to fucking fuck with me now. What else do we got?